apologize if my voice goes out. Another downside of being on the last day and having lots of great conversations. Today, I want to get you excited about oysters and in particular as a model system for looking at mechanisms that drive population divergence in the face of potentially high drain flow. For my dissertation, I've chosen the Olympia oyster, which is a marine bivalve. That was a little early. A marine bivalve native to the west coast of North America. So to set the stage a little bit, the Olympia oyster are alternating hermaphrodites capable of switching sexes up to 3 times in a year. And unlike some other oysters where both male and female spawn gametes, in the Olympia oyster, the females take in sperm and brood the larvae for 10 to 12 days, then release the weakly swimming larvae into the water column where they're there for 2 weeks up to a month. What makes this system particularly interesting is it's not entirely clear the effect that brooding has on limiting their realized dispersal. And so the Olympia oyster is patchily distributed. They prefer living in estuaries, and they're found from central coast of Canada down to Baja, California. Work done by my collaborator, Doug Ernestey, and others have found using mitochondrial loci a phylogeographic break at the Strait of Juan de Fuca from Vancouver Island and Puget Sound, separating oysters found to the south. Below Baja, California, there's a sister species, Ostrea conchaphylla. There's no observed morphological differences between the two, and they have in fact been combined again and separated again in the past couple of decades. And so my overall dissertation is looking at both the adaptive population structure in the species and the demographic population structure. So today is work for my first chapter using GBS loci to look at the fine-scale demographic population structure in this species. And so I'm a grad student at the University of Chicago, and there's obviously no oysters in Chicago. So in summer 2014, I went on a road trip of sorts and collected adult oysters from 20 sites, ranging from the northwest coast, Clasquino Inlet on Vancouver Island, down to San Diego. And then for my collaborator, I also have samples of the sister species, Ostrea conchaphylla, from 4 sites in Mexico. I assume most of you are familiar with GBS and RAD flavors of reduced representation genomic sequencing, so I won't go into it. The data set I'm working with now has 9,322 unlinked SNPs, unlinked referring to 1 SNP per GBS locus, and 148 individuals, including some from the sister species. This is after lots and lots of extensive filtering, in particular filtering for a minor allele frequency of 2.5%, and removing outliers identified in base scan and outflank. And so principal component analysis of SNPs just in the Olympia oyster, not in the sister species. On this plot, samples are colored by latitude, so warmer colors are to the north, darker colors are to the south, and the bigger circles are indicating the median value for each sampling site. And we see strong evidence of geographic structure. If we do a regression of latitude and PC1, have a strong correlation with a couple of interesting exceptions. So again, oriented north to south. These sites right here are my 2 sites from northwest Vancouver Island, which on PC1 are showing more similarity to sites to the south in Oregon as opposed to those in Puget Sound. Another interesting exception is, it's kind of a weird angle from here, is the site here, which is Coos Bay, Oregon, showing more genetic similarity on PC1 to Willapa Bay to the north. So to make it easier, I'm coloring these now by broad geographic region, linking again this Coos Bay, Oregon, and Willapa Bay, Washington to the north. And then as we go looking through other PC axes, we start to separate these out. And so on PC2 is separating out Vancouver Island, Puget Sound, and then Oregon and outer coast of Washington, as well as to a lesser extent, northern and southern California. On PC3, we're separating out Humboldt Bay, California, central California, and southern California. PC4, you get a separation between Vancouver Island and southern Vancouver Island and Puget Sound. And PC5, a separation between these two Oregon sites, and then Coos Bay, Oregon, and Willapa Bay, Washington. So looking at pairwise FST between sampling sites in those regions I just identified, you see a signature of isolation by distance. These are oriented south to north. So south to north and north to south, with again a couple of interesting exceptions. So this bright white square here with really low genetic divergence is between Willapa Bay, Washington, and Coos Bay, Oregon. They keep bringing this up because the population in Coos Bay, Oregon is known to be relatively recent. There weren't any oysters observed there 40 years ago, and they suspect that they actually came down with bringing in Pacific oysters, which is the current non-native commercial species on the West Coast, bringing those into Coos Bay, Oregon, and brought along some hitchhiking Olympia oysters with them. So it's exciting that this is recovering that signature and that story turns out to be true. So using tree mix, which is a model-based approach to show tree-like relationships between sampling sites. These are again colored by those same geographic regions and is outlining a similar regional population structure. And if we add in now the samples I have from the sister species to the south and root this tree and allow for migration events, the migration events tree mix recovers are events leading out from Puget Sound, which is this group here in orange, southward. So Puget Sound to Oregon, Puget Sound to Willapa Bay, Washington, and Puget Sound to San Francisco and Humboldt. I've gone down a little bit of a rabbit hole recently of reading fisheries reports from the 1900s when the species was primarily a commercial species before we ate all of them. And as the oysters native to San Francisco were over-harvested, they were actually sequentially bringing down oysters from the larger populations in Puget Sound and then even from Victoria and Vancouver Island. So I think this is, not to say that, I think this is recovering that signal of human-assisted migration. Again, to just test for a signature of isolation by distance, we do a Mantell test. This is with 10,000 permutations of values and show a significant correlation of water distance between sites and genetic distance. But another way that a recent method to visualize how genetic similarity varies across a landscape is EAMS, which stands for Estimated Effective Migration Surfaces. And this takes individuals that you have location information for and then calculates the observed genetic dissimilarity between them and then estimates migration rate and diversity among individuals. So for example, we have African elephants, forest elephants in green, savanna elephants in orange. We designate a habitat that they can be found and the number of demes or subpopulations represented by this grid. EAMS then assigns individuals to a deme and then estimates the effective migration rate across your designated habitat. So I'm going to orient you to this graph. We're going to see a few in a second. It's on a log scale here, with blue being areas of increased migration relative to the average, and orange being decreased migration And a decreased effective migration is in regions where genetic similarity is decaying quickly. And so my data set is kind of interesting compared to some of the other sets that Ames has been applied to, because with the exception of this little jutting out right here in Puget Sound, we have a very linear, almost like exactly typical stepping stone model of looking for isolation by distance. And so Ames working along this designated habitat identifies a couple areas of reduced migration compared to the average rate of migration. So on the left is just the overall results from Ames. On the right is areas where the posterior probability was greater than 90% that migration was above the average or below the average. Some areas that fall out in particular are around San Francisco Bay, and then around here at the Strait of Juan de Fuca and Willapa Bay, Washington. Also, I should mention that I've removed Coos Bay from this analysis because Ames was very confused, as Coos Bay had essentially zero FST with Willapa Bay far to the north. So it seems that Ames is recovering and illustrating the same phylogeographic break that has been identified by my collaborator using mitochondrial DNA, so that's exciting, but also a break or some area of quickly decaying genetic similarity centered around San Francisco. And if we include the sister species, obviously there's a lot of reduced migration centered from the sister species north, but we still recover these areas of reduced migration at San Francisco Bay and Strait of Juan de Fuca. As I mentioned, Ames also estimates the genetic diversity. Here with the same setup where orange is reduced and blue is increased, we see a latitudinal pattern of increasing diversity as you move south. And so looking at just expected heterozygosity, again, you get a strong correlation of increased genetic diversity with decreasing latitude. One of my hypotheses is from potentially ongoing or recent historical admixture and or introgression from the sister species to the south. And so that's something I'm investigating further with sequencing more individuals from more samples in the sister species to the south, as well as tests for introgression like F3 tests. So in conclusion, there's multiple lines of evidence for population genetic structure in this species on a regional scale. And the variation in effective migration rates suggests a more complex demographic scenario than pure isolation by distance. In particular, phylogeographic breaks observed at Strait of Juan de Fuca and around San Francisco Bay. However, all of these results are likely slightly confounded by the impact of human-assisted migration that happened very recently in this species' history. And consideration of this structure should be taken when planning restoration and transplantation projects. This species, as I briefly mentioned, in the early 20th century was severely over-harvested to the point where it was no longer commercially viable, and many populations crashed entirely. In the past decade or so, there's been a lot of renewed interest in restoring the species, both for its ecological benefits and as a potential sustainable aquaculture option in the future. One particular regulation that comes to mind that could be influenced by this is the restrictions on bringing oysters in from out of state to Washington are much higher than for moving oysters around within Washington. However, by seeing such a severe difference between the outer coast Washington species and those in Puget Sound would indicate that that might actually be of greater concern. And future directions for this work are, there's a new Olympia oyster genome that's being worked on, and so using that for improved genotyping as well as functional annotation of outlier loci, and using environmental data to detect putative loci under selection, given this species covers a great range of environmental variables and gradients. With that, I'd like to thank my collaborators and people who have helped me collect, and I have time for a couple questions, maybe a question.